All right, so this paper is titled Cerebrospinal Fluid Erection A Levels and Autonomic Function in KLS. So this is the first of, I hope, kind of a short series of kind of breaking down and summarizing um, some papers on KLS because there's a lot of jargon here that doesn't make sense to a whole lot of people without kind of walking through it. Pardon the animals, I'm at home. Um, So that's what we're going to do. We're going to kind of walk through it. All right. This is the first, so pardon me as I kind of have some growing pains. All right. Um, so the listeners of this kind of already know what KLS is. I'm not going to dive into that. Um, but I did want to point out kind of a couple a couple things. All right. So the authors of this are obviously introducing KLS, kind of why they're studying this. All right. So the goal of this paper is to essentially focus on whether orexin A or orexin or hypocretin, all right, that's a specific peptide that's released in the brain. I'll go into that a little bit more. But if that is associated with KLS episodes or exacerbations or relapses, all right, along with if there's an association with autonomic function, meaning the systems that we have in place that keep us breathing, heart rate, respirations, right? Those are examples of autonomic functions. Okay. Things that don't happen in our thought and control, they already do. All right. So that's kind of what these authors were looking into. So with that in mind, I just want to point out some words that might not make sense to the layman. Right? So functional imaging identifies thalamic hypoperfusion, thalamic activation, and or widespread hypermetabolism. All right, so the thalamus, that's what thalamic means. They're referring to the thalamus. That's a very, very tiny structure deep within the brain, just above the brainstem, that has many nuclei or collections of cell bodies that is kind of the master relay station for signals coming into the system and signals going out of the system. There's over 10 nuclei that are actually clinically relevant in the thalamus, from planning movement to executing movement to body sensory or somatosensory functions to hearing and vision. All those pathways that we think of going in and out of the body, they run through the thalamus. Okay. Hypoperfusion, what that's getting at is that's fancy term for exchange of oxygen and nutrients via blood flow. All right. Hypermetabolism, we all know of metabolism kind of in the nutrition sense, but hypermetabolism in terms of physiology, we're referring to biological activity of the cells. Some features are present in remission as well as relapse. For instance, thalamic pontine connectivity appears altered in and out of the disease state, with reversible deficits present in the dorsal pons. So pontine, that's just another way of us referring to the pons, which is a portion of the brainstem that is involved in executing, functioning, and relaying signals in and out, and largely different bodies of cells that function in terms of motor and sensory functions. Then they get into a little bit of an introduction with narcolepsy. Many of us have heard that. And this is kind of where we get the rationale, maybe, of the authors. In narcolepsy, the predominant theory is that there's a depletion or a lack of synthesis and release of the peptide orexin in the patients, producing or not keeping them awake. And that hormone is produced in the hypothalamus. And so, like the thalamus, and the anonymous like prefixes and suffixes, hypo just means below. And in this case, it's literally below and off in front of the thalamus. And that acts not so much as a relay station like the thalamus, but it has a ton of clinically important nuclei that are extremely minute, from being implicated in diabetes, because it has functions in the antidiuretic hormone, to temperature regulation, to sexual development with gonadotropic hormone release, to disinhibition, eating, hunger, our appetite, all of this stuff that we know of that is critically important to everyday life and functioning. There's probably, at least with respect to the brain, there's probably some level of interaction with the hypothalamus. So it's this tiny area that has all these collection of cell bodies that have all these different functions that are important. Okay. The authors wanted you to know that we have a twofold reduction in orexin, again, aka hypocretin, in a particular study that they found in narcolepsy patients. But sometimes it's conflicted, which they go on and highlight. And so again, the takeaway message outside of defining what some of those terms mean in the introduction, which is one of the goals of mine, so we kind of have a little bit more of a basis of what we're talking about here, is the takeaway message here is that the authors here wanted to focus on whether there's a specific brain chemical or peptide, aka orexin, which is aka hypocretin, to see if that was associated with episodes. Okay. And as we can see, we kind of can get a hypothesis that they believe there might be a drop in orexin, right? So what they did was they had some subjects. As we can see, they got approval for that. They had tens of subjects. So it's a pretty fair cohort. And they did some imaging, so CT, all right, that we all know as a CAT scan. Now we just call it CT. And along with MRI, pardon me, I'll mute myself, the cat's food's coming out. Okay. Both of which are not measuring function, they're measuring structure. So those are 3D, pretty much super fancy imaging that gives us a very detailed insight into the anatomy. HLA, that is the human leukocyte antigen. So that's directly involved in the immune system. Okay. We have a bunch of antigen presenting cells, and that goes into this whole other thing. But I believe that they measured that because there's a old study that showed a increase in the frequency of a particular allele for that certain HLA. All right, polysomnography and multiple sleep latency tests. So those are both versions of sleep latency tests. So they measure how much REM, non-REM, the specific waves, alpha, beta, gamma, delta, within sleep and when awake. And that's often used to diagnose narcolepsy. or rule out other conditions as well. Lumbar puncture, that's a common one that many know of. That's a procedure in which a very long needle, it's sometimes bigger, almost the length of your palm, that goes into the spinal column below our spinal cord in order to make sure that we're not striking the spinal cord in a particular space below our around the area of our third and fourth vertebrae in the lumbar area to extract cerebrospinal fluid, and that comes up and is basically like our plumbing system for the brain. So they measured through the lumbar puncture, they obtained the CSF, measured Orexin, all right, with a immunoradioassay. That's just a particular type of assay kit that they bought commercially for the lab in order to kind of standardize the limit in which they're going to deem as normal or low, which is what they describe here, their lower limit detection of what they would consider to be a significant enough decrease. All right. They also measured heart rate, 24-hour or heart rate and blood pressure, which is where they get into kind of trying to measure to see if there's an association with autonomic functions. All right. Data analysis. I don't know if there might be a single person that's going to listen to this that loves statistics, despite its direct relevance to everyday life. I know I didn't like it, but standard deviation, that just means how far away from the average that you are. Okay. A paired t-test, so they're comparing the same person to themselves just at a different time. So, for example, you compare a patient's orexin level while in an episode versus their own orexin levels when they're out of an episode. All right, so that's what a paired t-test means. A non-paired t-test, which is what they also did, is comparing the means of two different groups or two different cohorts. For example, comparing patients who were only tested while they were in an episode to those who were only tested while they were not in an episode. Two different groups. Right? Univariate analysis. So that's a very complex tool in statistical analysis, kind of trying to see if one factor, like being in an episode, leads to or causes a drop in blood pressure, to try to account, or even when you're trying to account for other factors. So that analysis is trying to eliminate some confounding variables. All right. The cosigner analysis, that is, again, a very mathematical way of trying to map out, or in this case, the body's 24-hour wave that it goes through, like heart rates, the rise and fall, to getting an average, low, high measure of that in these patients. All right. And what they found, all right, so again, they used 44 patients that met the criteria for hypersomnia. So something that when people are reading, they initially see in the methods 57 patients were identified in a given time period. But why did 44 patients only make it into the measurement? Well, there's exclusion criteria that needs to happen in order to mitigate confounding variables. At least that's the goal, is to mitigate as much as we can in order to make our data that is found to be strong. Otherwise, you can rip it apart if there's a ton of confounding variables that are not controlled for. All right. So that's the whole goal of all the methods and statistics and stuff that goes into various papers. All right. They found, and what they found was a roughly 31% drop in patients of orexin. All right, so it was statistically significant enough to confidently say that there was a drop in a good portion of patients. And in this case, they also found that structurally on the CT and MRI, there was not really any anatomic abnormalities, which is commonly the case for KLS patients. KLS is not a structural deficit or syndrome, but rather a functional one, which is why we often see that there's not anything that's going to show on traditional anatomic imaging. What else? So on the MSLT, or the multiple sleep latency test, they found that there was a significant number of patients that during relapse fell asleep extremely quickly. And this is what we refer to as delay, how long it takes somebody to fall asleep. And that was something that they noted in this paper, it being statistically significant enough to mention. All right. What else? All right. I'm kind of walking through this with you guys. I know that you see some highlights already. I kind of breezed through it, but I'm kind of going through it in real time. Okay, something that I apparently wanted to make note of is in episode, there were elicited symptoms of cognitive changes, changes in eating, depressive features, irritability, hypersexuality, and compulsions. And they summarized these into this table too. And something I believe the reason why I highlighted this was we all know the old triad of KLS from decades ago was hypersomnia, hyperphagia, meaning an increased or uncontrolled amount of eating, and hypersexuality, both disinhibited behaviors. And in this cohort of 44 patients, hypersexuality was only occurring in 8. That's 18%. Okay. Overeating, hyperphagia, the second part of that old triad, was only occurring in 4. So this is not directly relevant to the purpose of this, but I wanted to highlight it because the old triad that we used for decades in diagnosis, we're now having data within the last 10, 15 years that shows that that might not be the typical triad for a diagnosis that we need to have, because there's a lot of patients, and according to these numbers, most patients don't have hypersexuality and hyperphagia. Right? I believe that was the reason why I had that highlighted. So again, going back to what they were trying to get at, is that the CSF orexin levels, as I mentioned, were roughly 30% lower during episode than out of episode. That's what they mean by relapse and remission. All right. There was only 3 patients with the values recorded below the lower or proposed lower limit of normal. All right, so that refers to the methods that they used, the threshold that they were using to consider what was low and what was not. All right, although variations in the difference were noted, in each the level of orexin was lower in relapse. So what they're trying to get across is, in general, there was a significant enough of patients to report that there was a lower amount of orexin during episode compared to when out of episode. All right. Then they also measured heart rate and blood pressure, which I mentioned previously. And then what they were getting at here is levels of systolic blood pressure, diastolic, and heart rate. were all lower during the symptomatic state, or what they call relapse, or what we would call an episode during remission, or what we would call not in an episode. All right. And those were calculated over a 24-hour period, as we can see, and they show these nice graphs to show that. All right. And so again, what they're showing is they generally reported that the heart rate and blood pressure was lower during episodes on average than when out of an episode. All right. In the discussion, so in any paper kind of ever, we have the typical structure of introduction, methods, data, and or results, and discussion. The discussion is kind of where the authors kind of bring together these big picture ideas to talk about their findings and what they might mean. All right. And some of the takeaways really was that the authors found a difference, a significant enough of a difference in orexin levels that lined up with being in an episode than out of episodes. And while that is important to note, every study ever has its limitations. Okay, so it would be not justified enough in the scientific community or as any other reader to not read something and always take it at face value. That should not be the case. Even amongst the scientists, always critically assess something. And so I wanted to mention some of those maybe variables or limitations, as we would call it. All right. So for one, the heart rate and blood pressure. It is normal for there to be heart rate variability. And in fact, that is a measure of longevity. Healthy adults who have normal cardiac and respiratory function are supposed to have a drop in heart rate and blood pressure overnight or when they're asleep. That is supposed to happen. All right. So when somebody's in an episode and they're being measured when they're asleep, how significant enough and how much decrease in blood pressure and heart rate is actually contributed to some level of thalamic or hypothalamic dysfunction versus that normal expected drop that we do see when we're asleep. All right, so something that the authors could have done is measured those functions during episode while awake, because we don't sleep for 24 hours a day. We do still wake up to, whether it be go to the bathroom, take care of ourselves, to eat, drink, albeit we're incapacitated when we do so. Trying to take measurements as much as we can in those periods versus out of an episode doing the same things would have been a good idea to try to account for that confounding variable. Hopefully that makes sense. All right. As we can see, 44% of the patients, but not all of those patients had CSF samples taken. So could there have been some level of bias or some background story for why those 34, I believe, patients had their orexin levels measured compared to all of them? Maybe. And that is something that should be discussed more in the paper. Healthy controls. So what we mean by healthy controls or healthy matched controls is, say, somebody who's of the same age, non-KLS patient, roughly the same height, weight, not any other pertinent conditions that would confound it, have them as controls in the study. That's what we mean by healthy matched controls. And this study didn't really have that. They compared the KLS patients to themselves, which is great in terms of those paired and non-paired t-tests in and versus out of episodes. But how does that compare to the non-KLS patients? So those are some limitations that, from a critically evaluating standpoint, should be made known or should or warranted to mention. Now, what else would is there that I wanted to mention or maybe make note of? Orexin levels. Okay, yeah, something that they made sure to mention was that orexin is not necessarily specific to sleep conditions. However, there are other conditions like ALS, Parkinson's dementia, other neurological conditions that do have sleep dysfunction within it under their umbrellas. Orexin would not be able to be a really true biomarker for diagnosis in KLS or another condition because it lacks the specificity because it is involved in other conditions as well. That's what the authors are mentioning at the beginning of that. What else in patients? They mentioned histamine, right? That is involved in the sleep and wake pathways physiologically in the brain. It's another peptide, and by peptide, all we mean is a chain of amino acids that's more like a protein, like insulin. Insulin is a peptide. Contrary to, say, estrogen, that's a non-protein hormone. But histamine and orexin are peptides in the structure, and that they're not that they have a different structure than other hormones and neurotransmitters. Yeah, so basically it's saying here, it's just mentioning more and more background, like prostaglandin, like histamine, and amongst others that they did not assess in this, that may or may not be implicated in the function and or dysfunction of sleep. Okay. Now, what I wanted to go back, and so the takeaway that the authors found in this study was, with the limitations of the study that they have in mind and the way that they went on to do this study, their results indicate that for how they ran the study, they found amongst their patients that there was a drop in orexin levels in episode compared to out of episodes, and found a lower resting or on average heart rate, blood pressure in episode compared to out of episode. Now, I've been talking about orexin kind of indirectly, just as something that they've been mentioning, but I wanted to kind of hone in on what orexin may or not actually be, just for some further background. And so I mentioned that it's a neuropeptide. It has a protein-like structure, and it's synthesized in the hypothalamus. All right, specifically the lateral hypothalamus. So again, that is less than millimeters of particular nuclei in a certain portion of the brain that you almost can't even measure on you can't see on any imaging that we have in the particular nuclei, really. In that tiny little area, we have cells, hypocretin cells that synthesize this peptide. And so there's a couple of versions of it, and it kind of acts like the stabilizer, one of the several. We have other areas of the brain involved in sleep, like the suprachiasmatic nucleus. We have neurotransmitters like GABA and glutamate that are involved in sleep as well. Orexin is a peptide that is implicated in other conditions like narcolepsy, which is why they tended to pick this one to investigate. And without enough of it, because it kind of is one of those signals that tells the brain to wake up, without enough of it, you can see sleep, like in narcolepsy, and hypothetically, like in KLS. That's where that rationale came from. So again, like I mentioned, it's synthesized a couple with a few thousand. It's a very small number of well, relatively, it's like 10 to 20,000 neurons, which in the grand scheme of the brain is extremely small amount in the lateral hypothalamus. All right, so there's major players in the hypothalamus in terms of the sleep, which is why it's such an interest in KLS. And so what it's actually doing is that it helps to keep the brain alert by preventing us from falling into sleep during our waking life, really. And in the scientific literature, the system in which that's all involved in is the orexinergic system, if you want to look that up. And it's also involved in appetite. One of the nuclei within the hypothalamus that this is extremely close to, if not the same nuclei, then there's also nuclei that are directly involved in appetite, which is again something that we see changed in KLS. There's patients that have hyperphagia, and there's also a lot of people that stray away from their appetite. They don't eat, don't drink as much, right? And again, because it's involved in the hypothalamus, there is a level of autonomic control, meaning the functions like heart rate and blood pressure that we don't think about controlling because the brain already does it. So orexin is important in various functions, and particularly interesting in terms of sleep because it's implicated in other things. Now, it might not be, and it's probably not the whole picture. That's a reality check that a reader needs to see here, is that you can't take that at face value and be like, okay, well, I have KLS because I have a significant drop in orexin. There's many limitations to that hypothesis. And that's something that readers need to take into consideration. And so while, like, for example, there are other studies and there are cases, again, like I said, 31% of their cohort. Okay, so that means 70% of the patients did not have a significant drop in orexin. So there are a lot of KLS patients that have normal orexin levels with or without an episode. So that might be that there's somewhere in the pathway, not specifically with respect to the orexin, but something else at play. All right. So again, there's researchers that believe that KLS might be more so a disorder of the thalamus, like the relay station, in which if that's not functioning, then you have a disconnect between the brain, cortical structures, and function in the rest of the body. Such as like derealization, right? Not being able to perceive and be able to tell what's real and what's not, right? But because again, there's a lot of nuclei. Again, that just means bodies of collections of cell bodies of the same type. If the thalamus essentially closes the gate, it doesn't matter how much orexin the hypothalamus is pumping out if that signal can't get through. right? And so that's something that this paper can't distinguish. That's a limitation to our understanding of KLS and a limitation to this paper. So while it is a great paper, it's well done with some limitations in mind because that's just the name of science. Hopefully, this was able to help some people kind of break down and get some takeaway messages for what this paper, again, I'll scroll up for the name and authors, for what this paper was actually trying to convey and what they found and what some terms and jargon that are used help some people understand what the jargon might mean. All right, thank you guys for listening. Hope this helps.